All right, let's get right into it. Welcome to today's Explainer, where we are going to unpack the mechanism behind, seriously, one of the most transformative techniques in all of modern molecular biology. We're talking about polymerase chain reaction, or PCR. You can basically think of this as the ultimate molecular photocopier. It is the exact process that completely revolutionized how we study microscopic DNA because it lets us take a single, invisible genetic fragment and multiply it into something we can actually analyze, manipulate, and literally see with our own eyes.
So here's our quick roadmap for the session. We're going to define PCR, gather up our essential lab materials, break down the three-step mechanism, figure out how to visualize the results, and then finally look at some really cool real-world applications.
Section 1. What exactly is PCR, and how does it solve our microscopic DNA problem?
Okay, let's dive into the core concept. The formal definition of polymerase chain reaction is that it's an in vitro method, which just means it happens in a test tube outside of a living organism, used to amplify a specific target region of DNA. See, in the lab, we constantly run into this issue where DNA is just completely microscopic. If you want to sequence a specific gene, or say identify a virus, a single strand of DNA is kind of like a single needle in a massive haystack. PCR lets us take that one exact needle and duplicate it over and over again until it literally becomes the entire haystack.
And when I say amplify, I am talking about sheer unbelievable scale here. If your reaction is efficient, a standard PCR protocol takes just a few starting copies of DNA and turns them into over 1 billion copies in just a couple of hours. It's wild. Because the new DNA synthesized in one round immediately becomes the template for the next round, you get this massive, runaway exponential growth.
Section 2. Essential PCR materials. Let's gather up our molecular ingredients. Grab your notebooks because here is your literal grocery list for the lab bench. For a standard 50 microliter reaction, these are the essential reagents you're going to carefully pipette right into those tiny thin-walled PCR tubes. You'll need your template DNA, your Taq polymerase, forward and reverse primers, nucleotides, a specialized buffer, and sterile water to bring it all up to the right volume. But let's take a closer look at the true star of this show.
Taq polymerase. Now, you might be wondering, why do we use this specific enzyme instead of, say, a normal human DNA polymerase? Well, as we'll see in just a second, PCR requires extreme heat. A human polymerase would just completely boil, denature, and become absolutely useless. But Taq is isolated from Thermus aquaticus, which is a thermophilic bacterium that literally lives in boiling hot springs and hydrothermal vents. It actually thrives at around 70 degrees Celsius and easily survives the crazy heating cycles of the machine without breaking a sweat.
But here's the catch. Taq polymerase isn't actually very smart on its own. It needs to know exactly where to start copying. And that is where your primers come in. Primers are these short pieces of single-stranded DNA, usually around 20 nucleotides long. You need two of them, a forward and a reverse. They flank the exact region of DNA you want to copy, binding to opposite strands and pointing inward in the 5' to 3' direction. They are basically the starting blocks for our polymerase.
The last key ingredients are your DNTPs and your buffer. Think of the DNTPs as the raw bricks, the loose adenine, cytosine, guanine, and thymine nucleotides that Taq polymerase is going to stitch together to build your new DNA strands. And the buffer, which is packed with magnesium chloride, well, that's your mortar. It provides the optimal, chemically stable environment with just the right divalent cations to ensure the polymerase works perfectly without making a bunch of messy errors.
Section 3. The three-step mechanism. Let's look at the temperature cycle.
So, we have all our ingredients mixed in the tube. Now we pop that tube into the PCR machine, which honestly acts like a highly precise, programmable oven. The entire cooking process relies on a beautifully simple three-step cycle: denaturation, annealing, and extension. This sequence is going to repeat over and over, typically 25 to 35 times, and it is entirely driven by rapid temperature shifts.
Let's start with step 1 and see how this builds. Step 1 is denaturation, happening at a blistering 94 to 98 degrees Celsius. Now, in a living cell, an enzyme called helicase carefully unzips your double-stranded DNA. But in a test tube, we just use brute force heat. This high temperature violently breaks the hydrogen bonds between the nucleotide base pairs, forcefully pulling the double-stranded template DNA apart into two single strands. Next up is step 2, annealing. The machine rapidly drops the temperature down to somewhere between 55 and 65 degrees Celsius.
This sudden cooling phase is critical because it allows our tiny, highly specific forward and reverse primers to find their complementary sequences on that single-stranded template DNA and bind, or anneal, securely right to them.
And finally, we hit step 3, extension. The temperature bumps back up to exactly 72 degrees Celsius. And this is the absolute sweet spot for our extremophile TOC polymerase. It grabs onto those primers and rapidly synthesizes the new target DNA strand, adding nucleotides at a blistering rate of 1 to 2 kilobases per minute. Once it finishes the sequence, the machine cranks the heat right back up to 94 degrees, and the whole cycle starts all over again.
Now, if you are prepping for an exam or getting ready for your next lab session, definitely take a screenshot of this. It's your ultimate cheat sheet. Denaturation separates the strands at high heat. Annealing binds the primers as it cools. Extension builds the new DNA at 72 degrees. Just memorize this rhythm.
Moving on to section 4, visualizing the results. How do we actually know if this whole thing worked?
So, we've run our 30 cycles, the machine dings, we pull the tube out, and the liquid inside looks exactly the same as when we started. It's clear as water. Because again, our billion copies of DNA are totally invisible to the naked eye. So to fix this, we use a technique called gel electrophoresis. We basically load our PCR product into this gelatin-like matrix and hit it with an electrical current. DNA is negatively charged, right? So it gets pulled through the gel. The gel acts like a microscopic sieve, separating the DNA fragments by size. Because we made billions of identical copies of our specific target region, they all pull together into a distinct, physical band that we can finally see with our own eyes using a special stain.
Section 5. Real-world applications. Let's put PCR to the test outside the classroom.
PCR isn't just some abstract lab exercise you have to do for a grade. It is literally the backbone of modern biotechnology. It's used every single day to clone genes into plasmids, test patients for viral and bacterial infections, screen for inherited genetic disorders, and perhaps most famously, match biological evidence to suspects in forensic investigations using short tandem repeats, or STRs. And the crazy part? The foundational mechanism for every single one of these applications is completely identical.
So let's actually put your new knowledge to the test right now with a quick forensic scenario. Imagine you are working in the lab. You extract DNA from a single hair left at a crime scene. You run a PCR on a specific short tandem repeat marker. The result shows the perpetrator is homozygous for this marker, producing a single 200 base pair band. We have 3 suspects in custody. Let's take a look at their gels. All right, here are the gel electrophoresis results after we ran PCR on the suspect's DNA. Suspect 1 shows a single 300 base pair band. Suspect 2 shows 2 bands, one at 200 base pairs and one at 300 base pairs. Suspect 3 shows a single 200 base pair band. So, based on that 200 base pair marker from the crime scene, which suspect is our definitive match?
Boom. And this just brilliantly illustrates how incredibly clear PCR results can be. The match is definitively suspect 3. Look at it. Suspect 1 has the wrong allele entirely. Suspect 2 is heterozygous, meaning they have one copy of the 200 base pair allele, sure, but they also have a 300 base pair allele. Suspect 3, however, is homozygous for the 200 base pair marker, matching our crime scene sample perfectly.
Now, in reality, forensic scientists aren't just going to use one marker and call it a day. As our source material from Khan Academy points out, by combining multiple markers, scientists build a truly unique genetic fingerprint. In a typical forensic analysis, they'll use 13 different STR markers amplified via PCR. When you do that, the statistical odds of a false positive, meaning 2 random people having the exact same banding pattern, is less than 1 in 10 billion. That is the absolutely staggering power of PCR.
We've covered the primers, the polymerases, the intense temperatures, and the gels. But remember, this isn't just about reading a textbook. This is the literal code of life, amplified in a tiny plastic tube. So I want to leave you with this thought before your next lab session. If you extracted your own DNA today, pipetted in the master mix, and hit run on that PCR machine, what hidden genetic markers would you uncover in your own bands? Keep that curiosity alive, guys, and I'll see you in the lab.